Any student of cell biology can become fascinated by the way nucleolus behaves in the cell. You must know that nucleolus is the densest organelle situated inside the nucleus. It remains a dense body, moves a little bit inside the nucleus, that's all. But when the cell begins to divide and enters prophase, this structure disappears. And again, it appears after the telophase. So why does it disappear before the cell division? And then why does it appear afterwards? The question is that this structure is definitely very important for the cell. So people began to speculate on its function. Knowing a little bit about its structure and its composition, they were fascinated by the fact that it takes up cytoplasmic stain rather than nuclear stain being inside the nucleus. Well, things moved on, and cytologists began to examine different organelles in different cells of different animals. And a lot of people began to study oocytes, because in oocytes you find a number of dynamic activities. Mitochondrial biogenesis, ribosome biogenesis, and vitalogenesis. All these things happen in the primary oocyte. So people were fascinated by this cell and wanted to understand its implications and the role of different organelles in the formation of yolk. In doing so, one of these cytologists called Gatenby happened to examine an archaeannelid oocyte. Saccoceras was the name of this archaeannelid. And when he examined the oocyte of this animal, he found pyramid-like bodies arranged around the nucleus of the primary oocyte at a certain point. He went on and he found that actually the nucleolus becomes fragmented, and the fragmented nucleolus gets extruded, and once it gets extruded it forms a pyramidal body, the body like that of a pyramid, and the base of the pyramid becomes attached to the nuclear membrane, then the apex lies towards the cytoplasm. So you find all pyramids are arranged along the outer nuclear membrane. After a while, these pyramids get exploded and granules of yolk are spread out in the cytoplasm. In other words, the lipid granules are born inside these pyramids, and these pyramids are formed by the extruded nucleolus. So in other words, the nucleolus contributes to the formation of yolk globules. Well, he said that nucleolus is playing that role. So one function of nucleolus was established rather tentatively. But there were other cytologists who said that, well, no, the yolk is formed by Golgi complex. Some said that yolk is formed by mitochondria. So different workers had their own opinion about the nucleolus and the contributing factor of yolk formation. In the meantime, when the electron microscope was invented, it was found that the nucleolus is composed of three distinct elements: nucleolonema, a thread-like structure, a woven thread-like structure, somewhat helical, and just outside there is an amorphous structure, rather proteinaceous, called pars amorpha, and on the pars amorpha are attached or strewn a number of granules which are smaller than those of the ribosomes, and these granules were called pars granulosa. So nucleolus was composed to be of three parts: nucleolonema, pars amorpha, and granulosa, pars granulosa. Okay. That was all. But things went on from there, and the people had been studying oocytes in phase contrast microscope and electron microscope also. In 50s, the method for their isolation of a DNA began and became established, and that made a lot of people enthusiastic about isolating DNA from a wide variety of organisms ranging from bacteria to mammals. One of the instruments that proved to be an asset in those days was Model E analytical centrifuge. In other words, of course, ultracentrifuge. But this Model E analytical centrifuge is equipped with UV optics, so this lets you understand or examine bands of DNA spun in the cesium chloride density gradient. While examining those cells, the DNA of those cells, they found that there is also a band formed on either side of the main band of DNA called satellite DNA, and the satellite DNA, which was a heavy satellite, had DNA sequences that belong to Nucleolus. So Nucleolus actually was represented in the heavy satellite DNA band. Later on, it was found that the DNA which is contained in the Nucleolus contains sequences complementary to three classes of ribosomal RNA, 18S, 5.8S, and 16S, 28S, 28S, 18S, 5.8S, three classes of ribosomal RNA. In other words, the genes for these three classes of RNA are masked in the nucleolus. When nucleolus disappears, you may begin to wonder what happened to these genes. But in fact, these genes are present in one pair of chromosome at a particular locus called a nucleolar organizer. This particular locus is the gene. All the DNA are amplified 1,000 times, 10,000 times, and they are sequestered and brought into the nucleolus. In other words, the nucleolus owes its origin from the nucleolar organizer of the chromosome, and it contains the DNA which is complementary to three classes of ribosomal RNA. That became established by a number of biochemical experiments, hybridization experiments, etc. So nucleolus proved to be an indispensable cellular organelle. Of course, it is not bound by any kind of membrane, but it has DNA and some proteins associated with it, and it is the birthplace of ribosomes. In that, the ribosomal RNA are formed, and once the ribosomal RNA are formed, they are complexed with ribosomal RNA proteins, and these proteins are pro-ribosomes, and they ascend and they go to the cytoplasm, where they take part in the manufacture of proteins, the synthesis of proteins, because you know that the ribosomes constitute the platform where the protein synthesis occurs. The messenger RNA is attached, and on the one hand, and the transfer RNA comes and brings along amino acids, and they are joined by peptidyl transferase, and gradually the amino acids are linked together through peptide bonds, and the protein is formed. This thing happens in the ribosome. So the formation of ribosome is essential for a cell, because without ribosomes, you can't expect a cell to live for long. But protein synthesis must occur in all cells, because protein synthesis is vital for biochemical machinery. In order to run biochemical reactions, you have to have enzymes, and all these enzymes are proteins. If you by any chance happen to remove nucleolus from the cell becomes anucleolate, and that cell will not survive for long, because a cell cannot remain alive for long without ribosomes.